Hello, everyone. Welcome to a NanoString sponsored webinar that's part of the LabRoots Coronavirus Virtual Webinar Event Series. My name is Elizabeth Schneider. I am a program manager here at NanoString. Today we have the pleasure of presenting insights into the pathogenesis of COVID-19 as revealed by spatial biology. Our speakers today are Joe Beechem. He is currently the Chief Scientific Officer and Senior Vice President of R&D for NanoString Technologies based here in Seattle, Washington. He'll give a quick introduction to NanoString Technology. And then we have presenting the latter half, we have Dr. Chris Mason. He's an Associate Professor of Genomics, Physiology, and Biophysics at Weill Cornell Medicine. And as well, we have Dr. Rob Schwartz, who is a collaborator of Dr. Mason's, and he's an assistant professor of medicine in the Department of Physiology, Biophysics, and Systems Biology, also at Weill Cornell. You can pose questions to the speaker during the presentation. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question, and click on the Send button, and questions will be addressed at the end of the presentation. Thank you so much, our speakers, and now we will begin the webinar.
Thanks, Elizabeth, and it's fun to be here in front of a whole crowd interested in understanding the pathogenesis of COVID-19. I'm going to describe to you the technology behind the work that Chris and Rob did at Weill Cornell. Just to give you a little bit of an introduction, that's a picture of my lab there you see in Seattle, Washington. NanoString is a company of about 550 people. I got 200 people in R&D, and they did a lot of this work to help develop this technology. I'm really going to talk about two types of technologies that we developed associated with the COVID-19 crisis. You can see the GeoMx Digital Spatial Profiler is how we do spatial profiling of the rapid autopsy tissues that you'll be hearing from Rob and Chris. But we also have and developed technologies around the encounter analysis system, which is a non-spatially resolved system. And these are technologies we developed for profiling out of blood. So the technologies we developed are highlighted here. We're interested in molecular response, cellular response, tissue damage, and individual susceptibilities. And we're developing them for both bulk assays in blood and also spatial assays. Now, you know, the COVID-19, this is all about, you got to work fast here. This is a crisis. We had to put together a lot of technologies in a hurry. And, you know, how fast you can actually get speed to biological discovery is going to be a theme of this presentation for both me and Chris, because we need information and we basically need it now. When we started this, when this whole thing was breaking, actually, we were actually, part of NanoString was actually in Wuhan, China, and we really started in interacting with that group. I immediately set the R&D group in motion, and we actually started immediately working on tools we could develop for COVID-19, and we developed new gene expression panels that were what we call spike-in panels, and I'll show you what some of that data looks like. I assembled a team of over 50 scientists at NanoString, and we began an entire focused research effort. Our labs never closed down. And I'll tell you about some new technologies we developed, and you'll actually see them here. One of the other things we did is that as we were doing this, we started to get contacted by rapid autopsy programs around the world. And in these rapid autopsy programs, they really had to do discovery biology on fixed tissue. And if you're trying to do discovery biology on fixed tissue, the High-Plex technologies of the GeoMx system is pretty much the only system in the world that can accomplish this. And you'll hear from Rob and Chris, because they were one of the lucky first 10. We basically had to close it out. I got 4 microscopes running 24/7 on COVID-19 samples. You can see there at the bottom, on May 20th, I had to close out. We can't take any more. You can actually still interact with us with some specialized samples, and reach out to some of the NanoString people to find out how to do that. We have to go pretty fast, but these are the kinds of multiplexed assays that we're bringing up associated with COVID-19: host susceptibility, interferon responses, innate immune cell activation, tissue and lung function and impairment, the adaptive immune response, and then return eventually to homeostasis. If you're one of the lucky ones to come out of the other side of this terrible disease. Okay, and as we put these technologies together, you can actually see they expand greater than COVID-19. And we're interested in a lot of these other associated areas. Our focus today is going to be totally on COVID-19. Let me tell you about what we developed for the bulk profiling in blood. And one of the key things when we're developing this technology, if you're not super familiar with how NanoString works, we have an all-digital method to measure up to 800 targets at a time. And these targets, and these typically are gene expression targets. I think all the data I'm going to be showing you today will be gene expression targets, some protein targeting. But the key thing is that we can simultaneously monitor host and pathogen together in a very facile way. We're best in class in working with the tissue that is really the only kinds of tissue, safe tissue you can work with in COVID-19. And these are fixed paraffin-embedded samples for the spatially resolved work, and fixed cells and tissue, fixed blood samples also from blood, nasal swabs, and more. I think one of the key aspects of the technology that we've developed is that really one of the key things is you need your results and you need them in a hurry. So basically in 1.5 or 2 days, you measure 800 targets, you've got the biological information you want to capture, it works on fish tissue, and then you can spend all your time trying to understand the biology of COVID-19, and you're not worried about all that, how to set up your assay, how to do the measurements. And that's one of the key features of the way NanoString technology works. This is the range of sample types you can see on the right and the types of pathogens you can measure on the left. This gives you an idea how important speed was here. Okay, so this was actually one of the very first gene expression works that showed up in the literature at all on COVID-19. These were whole blood profiling using some of that technology that we rapidly developed in response to what was happening in Wuhan. And you can see that we actually went from this sort of an interesting example. This group, this is actually the Singapore Duke group, actually, the actual work was carried out in Singapore. They went from recruiting patients to a publication in 10 weeks. Okay, that means you have to have your technology. It's got to work out of the gate, give you high quality biological information. They were able to do that. It came out in Cell Journal in the host microbe literally 10 weeks after bringing in the first patient. This gives you an idea on what some of the things that we're measuring. These are blood profiling. And if you just look at those heat maps there, and these are COVID-19 fatalities there, and you can see 3 case studies on the upper right graph there. And you can probably just use your eyes. I don't have a pointer here, so you're going to have to bear with me. You can look at case 1, case 2, and case 3, and you see among the 770 genes that we were measuring the digital expression on, you can see the different color patterns associated with the genes associated with the response of those 3 different cases. And one of the first types of biology that came popping out of this particular study was the involvement of IL-1 pathways in some of the early, you can see the arrows there, some of the key markers on IL-1 pathways that are associated with the severity of the cases of the fatalities that were being measured at the Singapore Duke Hospital there. That was what was published on. I'm leaving the absolute results here. Hopefully, eventually, we'll be able to get you PDFs of parts of this study, but it gives you an idea of the biological information that we were able to rapidly recover with 10 weeks from patient in to this kind of information out, and we were able to publish on it. Now, shortly after that study, a larger case study showed up in medRxiv. I'm just going to highlight that. Again, they saw interferon type 1 responses. In this particular case, they were able to use our technology to profile 50 different patients in their case studies. And when you see that, now I can show you the heat maps of the responses. You can see these are much more detailed because we got many more cases. And you can see the no disease heat map gene expression case, the mild to moderate COVID-19, severe and critical cases. And you can see that each one of them has a unique gene expression profile that we were able to examine in the blood samples from these COVID-19 patients. And again, I'm not going to go through all the biology that's happening here, other than you can see there on section B of that graph. That's the classic volcano plot where you're trying to find biomarkers associated with these different key phases of the disease and criticalities of the disease. And down at the bottom, underneath the C, are the key overexpressed change gene expression pattern pathways associated with this disease progression. So he went from a 3-case study to a 50-case study in medRxiv, and then almost immediately after that, actually in May 21 of this year, published in New England Journal of Medicine was some of the NanoString technologies embedded into a case study that had enough information to make it all the way into New England Journal of Medicine. I'm just going to highlight what some of the gene expression patterns associated with that particular write-up of that scientific work. You can see a common theme here. You're actually doing COVID-19s versus various types of other lung diseases. In this case here, influenza A on one hand, and influenza, and that print's too small for me to see here, on another case. And you're able to see, actually it was the same. Influenza A is the normals, so-called normals, if you will. And then the two different sets of key gene hits that you see in the intersection of that Venn diagram are unique biomarkers associated with COVID-19 progression, both from sort of inflammatory pathway emphasis on one side and sort of like the tissue matrix elements on the right side, if you will. This sort of gives you a feeling of how this technology we developed to profile blood, and it gives you an idea. These are actually how we put these kits together. You can take some of this COVID-19 information and you can mix and match it with a lot of different features of the gene expression patterns, gene expression panels that we have inside NanoString. So you have a whole suite to choose from. These are some of the individual targets that we measured and monitored. These are the mix and match panels that you can use. So you can take this COVID information. If you're really interested in the inflammatory aspects of that, you can see that was in one of the presentations. You could actually take our inflammation pathways and mix it up with our COVID-19 panel plus, and that's how you or you can emphasize the innate immune response, the adaptive immune response, and you can do blood profiling on all these dimensions of the disease. We're actually making a 770-plex totally devoted to just this, and this will be out shortly. That sort of takes you through what I want to describe on the non-spatially resolved technology, and I'm going to bring you now into the technology we developed for the spatial examination of COVID-19 patient samples. As many of you know, in this era of technology development, we've been evolving from what used to be almost exclusive bulk genomics types of profiling here. And then in 2012, a very fantastic technology came along. Single-cell RNA-Seq came along and allowed us to get a lot of additional information. You know, the regrettable thing is you do all this single-cell work, and in the end, you need to know how those cells are organized in a spatially resolved manner, because these single cells don't actually do all the work. It's actually the tissues themselves that do all the work. And that was the emphasis behind developing this high-plex spatial biology technology called GeoMx. Now, this is how GeoMx works. And, and some of my talks, I actually have the slide that actually shows the original napkin where I scribbled out this idea. This was an idea I developed back in January of 2016. And it was all trying to address, how do you get quantitative high-plex protein and quantitative high-plex RNA information from fixed tissue, typical 5-micron tissue slices, FFP tissue slices, that represents biobank sample, which is really the currency of translational research. The key trick behind how we did that was, instead of actually inserting all of our assays into the tissue and trying to deconvolve them with colors or whatever, I'm never going to do 10,000 things at a time and deconvolve it out of a microscope. So what we did is turn the entire thing backwards. you take your affinity reagents, you can see little antibodies over there on the left side of the screen, or your FISH probes, and actually instead of putting colors associated with them, you put a sequencing barcode. It's a 60 base pair piece of single-stranded DNA, and inside that barcode, it says what analyte you're connected to. So inside the sequence, it tells me what antibody I'm connected to, or inside that barcoding sequence, it tells me what mRNA I'm connected to. Now, to make the thing spatially resolvable, we put that chemistry together onto those affinity reagents through a photocleavable linker. If you think about what do microscopes do for a living, they shine light in small places. So what this allowed us to do is literally go onto an FFP tissue slice, you can see on the right-hand side of the screen here, and we can light it up with 4 colors in a microscope. That's what microscopes do very well. But when I want to measure, and let's take Chris's example that you're going to hear about in a few minutes. If I want to take 2,000 mRNAs and I want to know what's happening exactly in that tissue for 2,000 mRNAs, I can actually go there. I will have 2,000 invisible mRNA FISH probes here. I go to that region of the tissue. I shine the UV light. All the little photocleavable sequencing barcodes would be released from that tissue slice, and you literally drink them with a microcapillary that sits right over the tissue slice. And then that generates what I call a digital replica of the affinity reagent as a function of space. So I'll just bring you through this in a big hurry here on the side. There's your slide. You hit it with your affinity reagents that all have these invisible photocleavable barcodes in there. You have a fluorescence morphology markers, we call these, allow you to see the tissue and visualize that. And you're going to see that from Chris and Rob's work. We go to an interesting pace. We shine a UV light. The photocleavable barcodes come off. We collect them, put them in a microtiter plate that's on instrument. We can cycle through that tissue about 30 seconds, collecting all these high-plex, multiplexed readouts, and that allows us to get this high-plex discovery information from these individual tissue slices. Now, I'm not going to have a lot of time to describe to you the details of how this works, but I will just a little bit. This gives you an idea what a tissue slice looks like inside the GeoMx DSP. And then when we profile, the profiling is by this UV light that we shined onto the tissue. And the UV light that we shine onto the tissue can take on very complex patterns because we built into the microscope 2 semiconductor chips, each about the size of half of your thumbnail, with 1 million mirrors on there. So when I want to do high-plex profiling of a unique geometry inside the tissue, in this particular case, say I wanted to monitor I want to do 2,000 mRNAs inside just the tumor part of that tissue slice that you see there. That would mean all those cells painted yellow. Those mirrors will adopt a shape that mimics that yellow stain on that tissue slice identically. We expose it to UV light, and we get a pure signal out of only, in this case, the tumor-specific cells. But when we get to COVID-19, we'll be doing things where we'll be looking at things surrounding the actual SARS-CoV-2 virus itself. And then we profile out of that. You can do rare cell profiling, and I'm going to gloss over that. We've had very good success with this technology. We've now had about 16 publications show up, back-to-back papers, and Nature Medicine was our very first work on this. A couple of covers there, clinical cancer research, a couple back-to-backs in Nature. So this is technology that's mature, it works. You get high-profile publications like this when you do these measurements. Now I'm going to describe to you how we adapted this GeoMx to become a pure discovery machine. And the way we did that is that we actually, the little photocleavable barcodes that you're looking at here on the FISH probes now have been, here's the photocleavable linker. You can see in that little circle there. And then off of the photocleavable linker, we put Illumina sequencing indices. That's where you can see the little I5 and the I7 dual index probes for Illumina sequencing. We got a universal molecular index in there that allows you to dedupe all your PCR library formation duplications. And then you got the unique GeoMx ID there. And it's the ID that tells you what affinity reagent you're connected to and what part of the RNA that you're examining. So in that kind of manner, it allowed us to generate infinite plex spatially resolved technologies. You can see over the last 2 years, as shown here, we've gone from 84 genes that we do in space to now routinely in the lab, I'm doing almost 21,000 mRNA probes in space. The work that Chris and Rob are going to show you is right there in the middle. It's around 2,000 genes in space that we'll be looking at, and Rob and Chris will show you. Now, I'm going to sort of gloss over a little bit, but I think the key thing I'm going to show you here is that your raw Illumina sequencing reads are processed with a pipeline that actually will link those Illumina sequencing reads back to the high-resolution images of the tissue you're examining. That's one of the key aspects of linking High-Plex NGS sequencing with the actual spatial imaging. And again, I'm showing you here, these are the molecular mechanisms we engineered into our gene panels that we examined the COVID-19 samples with. We work very closely with a company called ACD. It's part of a biotechnique company. And we develop visualization probes around these unique ACD visualization markers that allow you to go in there and see with your eyes where SARS-CoV-2 is, and then actually do your profiling around that. And actually, if you look at the progression, the virus titer actually, it's up for a while and it goes down in a big hurry. So you actually need more markers than just SARS-CoV-2 to do this kind of profiling. That's why we developed all these other things with ACD. We also developed an entire series of fluorescent antibodies that will allow you to quantitate unique aspects inside the tissue. This gives you an idea of some of the protein technologies that we're embedding. This gives you an idea, sort of a summary of the RNA probes that we'll be looking at for visualization and you have access to. So I'm now actually at the place where I can show you what this all looks like when you put it all together. So this gives you an idea on here. This is actually a rapid autopsy fixed lung tissue here. And on the left side, you're looking at the COVID-19 virus probe over here. You can see the dark red, and this is a blow up of a piece of that, the FFP lung tissue. And you can see over here, we've got the ACD probes now all working in fluorescence mode, and you can see how they parallel. This is a serial section off of the bright field. And now you can see how we're lighting up exactly the same spatial geometry that you can see in the bright field, except with fluorescence work, of course, you can have additional colors. So I'm actually lighting up TMPRSS2, and I'm actually also lighting up here ACE2 receptors, and we simultaneously allow you to visualize this. And then I can say, okay, look at this interesting region where SARS-CoV-2 is located. Let's probe what the immune environment is around this particular part of the tissue versus that. And that's the work you're really going to see from Chris and Rob. I can't show you a lot of stuff because even with Rob and Chris, they're going to have to be, we're discovering stuff nobody in the world knows. So we can't just actually be launching it here on a webinar and have everybody take off on all these hits. So I'll have to show you some of the work we're doing here. In this case here, non-human primate work with rhesus macaques. And these panels I'm talking to you about work fine in a non-human primate example. This gives you an idea of some of the visualizations we're doing. This is work we do with Harvard Medical School. We're working hard on this, and this is actually a work in progress. But the Harvard Medical Group allowed us to actually show you what this looks like pre-publication. This gives you an idea of the mRNAs that we're now spatially locating here. And you can look over on the left-hand side, the control region, and then you can look over here on the right-hand side, looking at regions that are virus high and virus low. These are all spatially resolved, multiple thousands of mRNAs we're doing in space. When you actually look and you look at what are the differential expression patterns now between the COVID-19 infected and then the control rhesus macaques, we can actually start generating volcano plots which show the differential expression in space of some of these key regulators associated with the COVID-19 progression. So if you look at that, I can't tell you a lot of these things, but they were nice enough to allow us to tell you. You can see the upregulated interferon response genes. There's MX1, IFIT3 that we're able to find in here. Cytokine pathways associated with STAT1 and CXCL10 are all upregulated in these particular tissue environments. And this is about all I could actually show you on those biomarker hits at this time. Yeah, I'm looking at my timer. I'm actually right on time for where I wanted to be. But hopefully I'm just sort of in summary to this technology development we did for both blood-based systems. We got some great results. This is things you can, a 2-day turnaround time, you get 800 answers. You can focus on what the biology of these COVID-19 patients look like at the patient level. And you can see we already got work that made it all the way into the New England Journal of Medicine. So you can really get high quality, high impact manuscripts out in this area. And then I switched and showed you all the technology we're developing for the spatial examination of these COVID-19 patients. And I took you all the way up to a non-human primate study. Now I'm going to have it handed off to Chris and Rob, and you're going to see some absolutely exciting work that the rapid autopsy program that the Weill Cornell has done. So I'm going to hand it over to you, Chris.
Great. Thanks so much, Joe. Really a pleasure to be on the call with you, and also Dr. Schwartz, with whom I've had the pleasure of starting a really fun collaboration with under very difficult times, though. But we've definitely been seeing a lot of new biology, and really excited to start to go through it of what we've seen. So I won't belabor the point that the outbreak we're all in the middle of sort of battling is one of the worst in human history, and it's still progressing and ongoing. And we really want to use some of the newer technologies to understand the basic understanding of the virus, its mutations, its changes, as well as its presentation in human patients, and also to look at it environmentally, look at it spatially, using some of the technologies, Joe, that you just described with some of the GeoMx and some of the spatial mapping, sort of new technologies that really give us a new window into biology. So in particular, I'll start, I'll describe some of the work we've been doing a lot on RNA-seq, as well as comparing it to RT-PCR and doing overall characterization of some of the clinical samples. We basically started banking and sequencing all the first batches of patients that came through the hospital, and I'm going to describe to you a result of basically the first 735 patients that came through, of which 216 were positive, the rest were negative, but I'll show you they did have some other sort of passengers. And we also did some environmental sampling and a good number of controls as well for some of these new samples. And so we're actually can start to compare some of the total RNA-seq from some of these samples to the spatial transcriptomics data, and really get a sense of what can we look at the correlation between what's in the NP swab or the nasopharyngeal swab and what manifests in the different tissues around the body during the course of infection, which, as you'll see towards the end from Dr. Schwartz, is also unfortunately led to autopsies. But from those donated bodies, we can actually learn a lot about the tropism of the virus and essentially what it's doing in the body. So again, I'll go over the RNA-seq, and I'll kick it off to Dr. Schwartz to describe some of the digital spatial transcriptomic profiling. And you can see here, this was some COVID patients, some flu, some non-viral patient samples, and then also normal lung samples. And again, using some of the latest panels you just heard described to really get an in-depth view of the tissues. But I'll start with what did we see in general looking at RNA-seq, and what can we expect for sort of the landscape of what the infection is doing in human cells. And actually, Joe had a few slides in some of the NEJM and other papers. But we can see we did a total transcriptomic profile of the patient sample. So this was basically looking at all kinds of RNA. And you can see here blue is human RNA, green is bacterial RNA. And you can see in the RT-PCR positive samples on the right panel the bursts of SARS-CoV-2 genome there in orange. And in some cases, we did get some false negatives from RT-PCR, but other cases, for the most part, it was spot on. And then we saw other patient samples that had almost as much as 80% of the RNA was viral RNA, actually ribodeplete the sample. So we could really see, this totality of the multi-kingdom biology, which is one of the things actually, I think it was at AGBT a couple of years ago, Joe and I were talking about this. And so I'm very excited to finally see it deployed now on the platform, because it is, an entirely novel way to learn about the biology, to think, cross-domain, cross-kingdom, and really look at all organisms that might impact how the body responds. And I'll get back to that shortly. But you can see here, we also can do a deconvolution algorithm from the RNA-seq to look at which cell types are present, and also you can do this, of course, with some NanoString data. But we can see mostly affiliated in goblet cells, and in some samples of subtle evidence of dendritic cells. But for the most part, it was affiliated in goblet cells that were predominant cell types in the samples we looked at. And there's another way to look at this. If you just separate them out with UMAP plots, you can actually see here the density by RT-PCR, normalized viral level, and also the predicted types of cells. So we see both cell types seem to be getting infected, at least at the site of collection for the NP swab. And you can see that across these different variables as well here. But the other kind of cool thing about sequencing it Because once you do the capture and actually then do, pull through and sequence it, you then can align to this viral genome and actually call genetic mutations and trace the evolutionary history of the virus. So we actually got from here, these are high quality assemblies and mappings. We can see some of the common variations. You see kind of these vertical striped lines, and also as well as more rare mutations from the viral sequence. And we kind of overlaid this with what's in GIS-8 for some of the larger global databases. And that's one of the most extraordinary things about this time is that any mutation we see, we can immediately reference and characterize relative to the global database and then understand it. So for example, when we started to see mutations that were even larger than SNPs, for example, this was a 9 base pair, 3 amino acid change that actually deleted a part of the conserved genome relative to SARS from 2003. We could actually see, this is a conserved portion of the C-terminal region of NSP1, which has already been linked to host chemokine dysregulation, which is exactly what we see. And we could take this data and then characterize, well, do we see this in other samples across the world? And indeed, we aren't the only ones. We can see from Iceland, from places in Ohio and England. We can actually quickly trace and see where are these other mutations appearing as sort of a matter of course. And so that's, not too surprising because we're sequencing the virus, but I think what's also really inspiring is to think about what else you can learn about the biology when you have probes or you have sequences coming from other viruses. So in particular, what's been done for the Seattle flu study, for example, and Trevor Bedford has an amazing Twitter feed about this if you haven't seen it, is, every year they've been looking for other viruses as they burst up in the winter season. But now we can also do this with total transcriptome RNA sequencing. So in this case, we look to see for low, medium, and high viral levels, you can see here in the middle, or was it not detected? Or over on the right, we look to see, did we actually have evidence of other viruses, other respiratory pathogens that might be present? And you can see over here, we saw co-infection within the SARS-CoV-2 patients that were positive was relatively rare, but we did see pneumonia, we did see other influenzas, other rhinoviruses, other viruses present, other RNA viruses that are present in these samples. And so you can see, in general, once you're SARS-CoV-2 positive, that's what's driving a lot of the biology. But the interesting thing is when we look at the microbiome as well from this total RNA-seq data, we can actually see again that none, low, medium, and high patients, and these are normal flora in airways or oral microbiome are shared. And we can actually see that there is a little bit of a disruption, a microbial disruption in these high viral titer patients, indicating either the patients were susceptible before they went in or that something after infection had displaced and actually disrupted the microbiome density for these generally commensals. So that's another interesting feature we've seen in the data, and we're starting to look more in the GeoMx data sets. And then also echoing back what Joe had mentioned before is we do see the human response in the RNA, which we can see some of the exact same genes you just saw in the previous slides as some of the interferon-stimulated genes or interferon-related pathways you can see really strongly upregulated. And in particular, actually, if we look at the pathways of these, the ones that really get upregulated are the interferon response, antiviral modules. But also down here, we can see disruption of the olfactory signaling pathways, which we see more pronounced in some patients that have lost sense of smell or taste, which is really intriguing to think about what that means clinically. And also the hemoglobin complex, and also looking at iron binding and other oxygen binding pathways that relate to some of the hematological deficiencies and disruptions that have been seen in patients as well. And maybe even linked to some of the happy hypoxics that have been seen clinically, but although that's speculation at this point. So the other thing we want to do is look at basically cytokine and interferon signaling, and we can see here IL-6 and other cytokines and other inflammatory interleukin markers really disrupted, especially in the high viral titer patients you can see. And we've also, this is some figures from our paper that's online, but we've also made a website where you can go and make some of the same plots yourself, which is called covidgenes.weill.cornell.edu. And it's a place where if you want to plot some of the data from this study, you can actually go there and start to play with the data, and built it basically as an online resource for anyone in the field trying to learn about, when you see disrupted expression in your samples, you want to learn about the expression data, you can go here to play with it. And we're going to be actually, now we've got all the spatial data, we're going to start to upload some of these data as well to actually, I think, add this as a nice resource for the community. And the last few things I'll close on is just we did observe also, getting back to some of the clotting and sort of blood problems observed in some of the patients, we could see complement factor pathways are disrupted also as a function of viral load. And then finally, we tried to link to the clinical metadata to see if we could learn anything there. And we did see, for example, in the high viral titer patients, a high level of ACE2 expression that was generally commensurate with the amount of virus. And we looked to see clinically, did this change the risk of infection, which the answer is yes, you can see on the left here. Not necessarily mortality, but a little bit of risk of intubation. But we did see that really this is a function also of hypertension is driving us a lot to some degrees, driving us a lot. So with or without ACE inhibitors or angiotensin receptor blockers, ARBs, both of them didn't fare well, when they had hypertension. So we think, interestingly from the data in the hospital records, it does have some risk of a higher infection, but not necessarily mortality. Hypertension is actually what's driving this more than anything else we've seen. And so that's, there's a lot of open questions. I think, what are the stages that are present and as a function of the virus during the time of infection, how does it move through the body? And I think that's what Dr. Schwartz will get to next, to think about what we can visualize. And I think, that's kind of I think I'll hand it over to Dr. Schwartz to start to get into some of these questions.
Well, thank you very much for that segue, Dr. Mason. And I just wanted, again, to thank the group here for the opportunity to be working on this project and how excited this work has been. And so with that said, and it's been mentioned by Joe and Chris, there's a lot of open questions here with COVID-19, which has really engendered kind of a lot of the excitement here in its study. So, in kind of the intersection here of, some of the types of problems that we're seeing in our patients and how we can address it with these technologies, some of these questions are, like, first, are there different or distinct stages of COVID-19 induced lung injury or disease? Does SARS-CoV-2 virus-induced lung injury differ from other viral infections, such as flu? Does SARS-CoV-2 induced lung injury differ from other ARDS or acute respiratory distress syndrome or lung injury type of processes? What cell types are present in the COVID-19 lungs as compared to normal lung, an injured lung? And is the spatial organization of cells in the lung altered in COVID-19 disease? And so basically, to try to address this question, we were actually able to leverage the spatial transcriptomics platform using the GeoMx. And so basically the idea here was to collect a variety of lung tissues, which Chris had already mentioned earlier, from basically patients who had either died from COVID-19 related acute respiratory distress syndrome or ARDS, influenza related ARDS, basically non-viral causes of ARDS, or just normal lung tissue. And basically we're doing this to try to understand the similarities and differences in host responses to ARDS in each of these settings, and also examining kind of what was going on within the tissue in a spatial context. So we basically examined, at least to start here, at least 3 patients from each of these different conditions, and these all were from FFPE samples from these patients. And that's also important to note, because one of the challenges in studying COVID-19 is safety. And so all these samples have been processed in a way which eliminates any active virus. And so these are all safe for us to work with. And so these were profiled as already described by Joe. So, obviously we're here in New York, and obviously these samples were all sent to Seattle, to NanoString. And within the tissues, we actually picked basically a variety of different regions of interest. So from the large airways, the vascular zones here, the alveolar zones, and then also the macrophage infiltrates, which were denoted with that CD68 staining as described earlier by Joe. And so to kind of give you an idea of what this looks like, you can kind of get this here. And so this is normal tissue, basically, and you can kind of see here the large airways, the vascular and alveolar zones, and you can see that the tissue structure is very nicely maintained, because this is coming from more normal tissue. And essentially, each of these specific cell types were labeled, and we can identify each of the regions of interest that were picked within this. So if we actually look at COVID tissue, there's, first of all, some damage that obviously that's occurred to the structure of this tissue, but each of these regions can also be selected as seen. Now, basically the profiles, the tissues were processed as already described by Joe through the processing as delineated here, and each region of interest was essentially illuminated and basically put into the GeoMx pipeline. And so what you can see here, sorry, that went a little too fast. Basically, there are a variety of areas of interest that were processed. Some of them were removed just during quality control, just because they didn't have enough cells or enough individual genes that were detected. But you can see here that we do obtain a lot of areas of interest from the alveolar zones, and then a smaller group from these large airways and vascular zones. And these were crossing across all the different tissue types here shown on the right, you know, ARDS, COVID lungs, flu, and the normals. So basically, obviously, we don't have to necessarily go into the specific you know, genes going across here, but what you can get an idea is that there's a variety of genes that were pulled out from this array, including the spike-in genes from the specific COVID genes, and even with some of the genes that were moved from the lower quality cutoffs, we still had a lot of genes that were still able to be assessed across all these different samples. And so if we actually now start digging more into the data that we're seeing here, is that one of the things that was actually quite surprising to us is that the normal lungs actually clustered quite closely with each other. But one thing that we were surprised was that, you know, we had ARDS from non-flu and then ARDS from flu, and we expected these to actually be, you know, it's ARDS, but actually they cluster quite separately within the analysis, and that's one thing that came out here. The other thing was that the COVID lungs actually clustered quite also separately from each other, basically kind of capturing that even though you know, these are all patients who ultimately died from COVID, there's actually obviously quite significant heterogeneity in the different disease components that we're capturing here. This is also reflected somewhat in the differential gene expression results that we're seeing. There is heterogeneity among the COVID lungs, but with that said, there's pretty striking differences that are present within the COVID lungs as compared to the flu lungs, COVID lungs compared to the non-viral lungs, and COVID lungs compared to the normal lungs. And I know I'm not drilling down here deeply into each of the specific genes that we're seeing here. There's a lot of commonality that we were seeing between this and across all the different COVID samples. And this is denoted here with the essentially the signaling pathways that are upregulated here. You can see here, just on top here, that the cytokine signaling here is markedly upregulated, which is something that Chris had already pointed out earlier, a variety of interleukin signaling, TNF signaling, and others. And this is all consistent with what we're seeing here clinically and as well as histologically in these samples. So it's really capturing that quite well. And as we drill more deeply into this over time, we're hoping to be able to better understand the biology here that is going on. Now, one of the technologies that can be deployed is just quantitative single-cell deconvolution, and it kind of gives us an idea of the different cell types that are present here. You can see here that within the alveolar zones, we're obviously capturing a lot of the epithelial cells and some of the immune cells as well as the blood cells. But as we go through each of the different ROIs that we examine, we actually are capturing the representative cell types as exemplified here with the ciliated cells. So, it really, again, gives us an idea of some of the dynamics that are going on here, and that's really exemplified here where you can see kind of the differences between the different subtypes or samples here with ARDS, COVID ARDS, flu ARDS, and just the normal tissues. And I mean, basically, we are trying now, this is kind of early times, but we're really going to try to use this data, try to understand the different cell infiltrates that are being driven in the context of the disease process and how this may be ultimately impacting kind of on the histology that we're seeing and also the downstream signaling. Now, this is kind of looking at more the bulk analysis, but we can actually look here within the specific sections themselves and see where the different cell types are being mapped spatially within the sections themselves. And, I think this is one of the real strengths of this platform here. You can see, macrophages, monocytes, neutrophils, fibroblasts, DCs, and T cells. And this is just the different cell types that we're showing you here. But you can actually see that in the different disease states that they're actually quite different. And if you actually just drill down here in the COVID samples, if you look here on the left in the macrophages or monocytes, is actually, if we link this now to the clinical metadata, which we just don't have enough time to discuss today, this actually correlates with the different stages of the disease that's going on in the patients. And so this is going to be really insightful as we link this to the transcriptomic profiles being generated from each of these spatial sites. This is just, sorry, I forgot we had this right here, but this is just zoomed in here for the macrophages right here. And so, I mean, this data has just been very exciting to correlate with what we've been finding clinically and with the clinical metadata. Now, we can also cross this with the RNA-Scope technology available with this, and basically looked at a variety of different markers, basically looking at the SARS-CoV-2 spike, ACE2, and TMPRSS2. And this just kind of gives us an idea of the different level of expression of these different markers here. And I think this somewhat reflects, when you see some of the loss here, just the overall global extent of damage and loss of some of the cell types that would be expressing this at these different stages. But this is just giving you an idea of what this looks like. In one of the patients, basically with large airways, had a lot of the TMPRSS2, but very little that were present in the alveoli. And basically, this is a patient that had very significant alveolar damage because of the stage of illness that they had. COVID-19 is in a later stage of the illness, and we still then saw maintenance of TMPRSS2 throughout the tissue, but a lot of loss of the ACE2 expression, and that's just reflective of the cell types that were being damaged or lost in the course of the infection at this stage in these patients. And you can see here, it's not necessarily important, the specific patient information, but just the differences as how this is reflected in the patient clinical presentations. So, I mean, I think what we would be able to conclude here, and just as a summary, is that the clustering and differential gene expression results show that the samples are quite heterogeneous within the COVID and ARDS groups, but actually quite similar within the normal and flu groups, which actually, I mean, the normal made sense to us, but the flu groups we actually expected to be more heterogeneous, but actually turned out to be more homogeneous, and that might give us a little more insight into what's going on in the COVID disease. RNA-Scope technology can be leveraged to specifically look at a variety of different genes. Specifically here, looked for TMPRSS2 and ACE2, and the quantitative single-cell deconvolution actually can be used to show the specific cell types that are in the COVID and ARDS groups, and they're actually more variable, again, as compared to the normal flu groups. So I'll let basically Chris just basically show off his group here, who have obviously contributed to the group here, and I'll just let Chris, you can hop on there and do that, and then I'll hop on in a second.
Hop back up, yeah. Great, awesome, Rob. It was really great. So it's been, then we've both been really excited about the data that's coming in. So just thanks to funding from everything from the NIH to different foundations such as World Quant and Citadel, who helped this, as well as Gates Foundation. Thanks to collaborators, especially obviously Dr. Schwartz here, and everyone is also at Cornell and at NanoString has helped get this set up, and then pitch it back over to Rob here for his thanks.
And I just really want to also just acknowledge the funding agencies, which are the DoD, the NIH, and some basically the Irma Herschel Trust, and then also the people who have done this work in my lab, denoted here in red, and former group members who have also contributed to this. But the most important is just also to credit Alan Borchik and the rapid autopsy group and pathology group here at Weill Cornell and New York Presbyterian, which really we've worked very hard together to build this database and library of material, and it's really been due to their heroic efforts here. And so with that said, I will turn it back to the moderators here so we can address some questions.
Thank you so much, Joe and Drs. Mason and Schwartz. We really appreciate the presentation. It was very interesting. So as I mentioned in the beginning, feel free to reach out with a question through the chat. We did receive some questions throughout the presentation, so I will go ahead and address those now. Please don't fret if we don't get to your question in the Q&A. I believe the folks at LabRoots will be collecting the questions, and hopefully we can follow up with you specifically to address those questions after the virtual event is over. So the first question is for Joe from NanoString. So, why are nCounter and GeoMx technologies especially well suited to study infectious disease and COVID-19? And part of the question as well was asking, what makes the nCounter and GeoMx technologies more practical than, say, doing like RNA sequencing?
Hey, great. Thanks, Elizabeth, and thanks for the question. I think a lot of this comes down to, one is that, and I think both Rob and Chris highlighted this, is that, these are dangerous materials, so we really have to be careful, and our hats off to the rapid autopsy scientists down there doing that work. It is incredibly dangerous work, so we need to fix these tissues and we need to fix these blood samples so that, we don't sending coronavirus through the mail up to Seattle and all my people getting what is happening in New York and vice versa. So, and now one thing to mention, I've been running R&D at NanoString for a number of years, is that we believe fixed samples are so crucial for moving forward translational science that, basically everything we do at NanoString has to work on fixed tissue before, or else we just don't develop it. So both the nCounter technology and the GeoMx technologies are totally at home in fixed tissue. And, by advancing this technology to the place where we have essentially infinite multiplexing now with the NGS readouts, you can now do pure discovery on things that were normally never pure discovery kinds of tissues. So I think that's the essence of it, Elizabeth, is we can work with the right kind of tissue type. And then the one other key aspect is that you want the scientists getting to the biological information as fast as possible. You don't want to be waiting for weeks for something to come out of some pipeline or some bioinformaticist that's gonna help you out. You really want to return the information to the scientists so that they can really start paying attention to the biology. That's what matters, not the assay. So you really, and that's something we worked really hard on too. I think it's those two factors, Elizabeth.
Great. Thanks, Joe. Another question that came in also for you is, is there the ability to make custom assays for gene expression on nCounter as well as custom GeoMx protein and RNA assays?
Yeah, thanks, Elizabeth. Thanks for the question. It's absolutely essential. I think, a lot of companies don't do custom things, and it's like, well, you've got to do custom things because there's going to be information that Chris knows and Rob knows that nobody in the world knows. And when you want to be able to probe those types of things. So, yeah, custom reagents can be made for the nCounter as sort of our standard business. So you can custom all the way up to 800 customs if you want. On the GeoMx, it's similar kind of thing. You can have custom probes come in, just like we did a custom collaboration with ACD to allow those visualization markers. You can pick your special target of interest, and custom probes can be made for that. And they can be mixed in directly with these panels so that we reserve these barcode regions just for custom types of additions. I think it's really important. Thanks, Elizabeth.
Excellent. So we had a couple questions that came in for the latter half of the presentation for Drs. Mason and Dr. Schwartz. So how does the cellular composition of these rapid autopsy samples differ between the different disease states like COVID versus influenza, and then, also just normal lung tissue?
Yeah, I guess I'll jump in. This is Chris. We do see distinct, there is for sure some overlap on the, interferon-stimulated genes, the response to the infection, some of the same pathways. but also there are unique ISGs, there are interference-stimulated genes that are either distinct or even much more strongly upregulated in response to COVID-19. So it's definitely not, quote, just the flu, as evidenced by the molecular data. It really is a much more disruptive and also disruptive in distinct ways, especially in the, in sort of IL-6 markers and also some of the just general inflammation stress on the body.
I mean,
Thank you.
I would just add to that. I mean, basically, one of the things that we've been seeing is that, obviously the timing of the infection, of where we're getting the tissue matters. So earlier on, the composition, appears to be a more, neutrophilic type of composition. But as the disease progresses over time, we see a much more macrophage infiltration. And I mean, I think that's reflected in what was seen on the GeoMx analysis. And, I think what Chris just mentioned is what we're trying to understand is how is the signaling driving the recruitment of different cell types into the lung and how that's contributing to some of the downstream effects, injury, and things that we're seeing. Thank
you. So a few more questions. So you guys had done some RNA sequencing, looking at whole transcriptomics of some of these COVID-19 samples, and then you did the GeoMx in space. So what was the concordance, if any, between the the RNA sequencing data that you did in bulk and then the GeoMx data that you did spatially?
I guess I'll go first. It's actually been quite beautiful. We see some of the exact same signatures of the genes that you can pick up at the site of infection, really propagating through the other sites of the body that either had infection or have it currently. And you can see this interesting, really it's a systemic disruption of some of the same genes and pathways. And it also just leads to you can see the immune system coming in and getting, really trying to do what it can with the disease, but obviously in the case of really advanced diseases, struggling to even keep up. But we actually don't have an R squared to say. We just got all this data, some of it like yesterday. So we'll be doing a lot of those calculations now. We look across different sites and across different cohorts. What's the correlation? How predictive is one versus the other? But we're looking at that now.
Yeah, I would echo that. I mean, from we actually from the autopsy samples, which obviously is looking at a slightly different state, which is at the end point here. But what we found is that it actually correlates quite closely with what the GeoMx was showing. And basically, the one thing though that obviously GeoMx is adding is kind of the spatial information, which we're losing in the bulk analysis.
Great, thank you. So just a few more questions. So do you plan to do this profiling in other types of tissue as well? I mean, you mentioned earlier that, this virus affects different tissues in the body and could affect it differently. And as a second part to that question, do you have any data on any of the comorbidities of these lung tissue samples from these different patients?
So we have all the clinical metadata from all of these patients, the comorbidities that they have, age, gender, medications they're on, obviously how long they've been in the hospital, and so we have all that data. And basically, to answer the question, do we plan on running additional tissues? That's something that we're, obviously we first focused on the lung tissue because obviously that's the primary site that's leading to the death and endpoint of these patients. But a lot of the other tissues that we've been examining on autopsy have been showing very profound and unusual types of injury that hasn't necessarily been reported in other disease states. And so, I think there's definitely a lot of interest on our side to examine these tissues with these types of technologies and approaches.
Excellent, thank you. So before we wrap up, I myself as the moderator actually have a question. One of the things that we are seeing with this particular virus, as you mentioned, is sort of the degree of havoc that it wreaks on, different tissues and on, the vasculature, the gut, CNS, etc. And some of the initial data, and Joe highlighted this in the first part of the presentation on the nCounter side, is suggesting that, you know, it's not actually just the initial infection and the virus that's doing the most damage, but it's really the inflammatory response that is kind of going into overdrive and is, you know, causing some of this tissue damage and organ damage. So I was wondering if you could comment on that in the context of this particular study and some of the other things that you've seen just in general of studying, you know, SARS-CoV-2.
I mean, I think it's, you know, it's a very important question. You know, basically, you know, what I would say is that one of the things that we've been trying to capture and understand is the different stages of the lung disease, which, I mean, right now you know, one of the exciting things is that everything we are learning is new. But one of the issues has been, obviously, we don't fully understand the disease progression as it exists in this context. You know, my belief is that that there's different stages of the disease, one where the virus is much more active and prevalent, and then, as you mentioned, kind of this havoc stage, which may or may not be related at that point to active viral replication and high viral titer. And so, you know, I think the way to address this and what we're hoping to do is try to partition between these different groups by getting, you know, more samples, more data to try to better understand each of these disease states and how they relate to each other and how the virus may be driving the latter havoc outcome, as you put it.
Great, thank you so much. So that wraps up the Q&A portion of the webinar. Again, if you didn't get your questions addressed, please go ahead and submit them through the chat, and we will be collecting them at the end of the presentation, and we'll follow up with you. Thanks to our speakers again. It was a very insightful presentation, and thank you all for watching.
Thanks.
Thank you.
Thank you, Elizabeth. Good luck on your research out there, people. Bye.
Stay safe.
Thank you, guys.